Chapter 46, Microbiology and Disease. All right, so microbes are everywhere, even in the air. And of course, before the discovery of the microscope, they didn't know that there were little things that cause pathogens. Right? So some of them will cause infection and disease. And for the MA, your role is to be able to identify the microbes. That way we can treat them properly, treat infections properly. We'll do a few collection techniques, and then we'll talk about testing and viewing these specimens. All right, microbiology, the study of microorganisms. So we'll talk about mostly single cell organisms and subatomic particles. The normal flora are the natural bacteria, fungus, and virus that are on your body. And a lot of their role is to prevent the pathogenic microbes from attacking your body. So if you kill off your beneficial bacteria, then those pathogenic ones can take hold. So for example, if you use a inhaled steroid for asthma, that's going to kill all the healthy bacteria on your body, and you'll be susceptible to fungal infection. Role for the MA in microbiology. So definitely ID. Obtain specimens, examine them, and or send them out. So we'll do a lot of microbiology procedures. All right, so we'll talk about the different types. So we'll first talk about subatomic. Subcellular, I should say. So a virus is not a complete cell. It's actually smaller than a cell. Localize or become systemic. So this is like a skin infection or where the scar is, whereas systemic, it might be in the blood or the cerebral spinal fluid. How do we classify microbes? Well, the structure. So I mentioned subcellular, like virus and pyrons. So have you ever heard of mad cow disease? So mad cow disease is not caused by something cellular. It's actually caused by proteins, a pyron. And then for viruses, they're not a cell, a complete cell. They're smaller. Virons and pyrons. Next we have prokaryote. Pro means before. Karyote means nut. So if you take a look at an avocado, the seed looks like the nucleus. So prokaryote is before the nut, whereas eukaryote is a true nut. So prokaryote, no nucleus. Eukaryote, a true nut, will have a nucleus. So the DNA in bacteria, prokaryotes, they're scattered throughout the cell. They're not localized within the nucleus like our human cells. Standardized naming. So you guys are probably familiar with E. coli, right? So that is the genus name and the species name. So for the genus name, it'll need to be capitalized. The species name is lowercase. So for us, we are Homo sapiens for humans. H. sapiens. Genus name, species name. So capitalize. So here we have staph. Staph means it grows like grapes. So the word staph means bunches or grape-like. And coccus means round shape. So here we got a bunch of round balls. And then here we have the species name. It tells you further definitions. Have you ever heard of a staph infection or MRSA? It's Staph aureus. Or it can be on the skin, Staphylococcus epidermis. Describes where it's located. Ooh, question 2 first, I guess. Describe the classification of organism and give an example of each. So, what are they asking for? Subcellular, prokaryote, and eukaryote. So subcellular would be virus. Prokaryote would be bacteria. And then eukaryote would be fungus. There you go. So virus, subcellular. Prokaryote, bacteria. And then everything else, protozoan, fungus. Parasites are multicellular. So we'll call those infestations, whereas these would be infections. How do microorganisms cause disease? What do? They steal your nutrients, cause toxins, attack the body. All right, viruses. So pyrons and virons. So we won't talk too much about pyrons. So the only representative disease of that is mad cow disease. So if you ingest certain proteins, then it'll cause a disease. Kind of weird. Viruses, super simple. All they are is a protein capsule and some kind of genetic material on the inside. It could be DNA, RNA, single cell, or double-stranded. That is it. So they can't self-replicate. They have to actually inject into a host cell and hijack that host cell's mechanism for replication. Because of that, it's very hard to grow viruses in the lab because you'll need susceptible host cells for them to attack and multiply. Some common viral infections: HIV, human immunovirus, and hepatitis. Pick a letter. We have A, B, C, D, and now a bunch of other ones they have identified as well. Who's at risk? So for hepatitis HIV, intravenous IV users, those who practice unprotected sex, they're at higher risk for these infections. HIV is the virus. So once you contract a virus, you can never get rid of it because a virus can hide. Once your T cell count is below 200, you now have the syndrome, the acquired immunodeficiency syndrome. So the virus has attacked your body, damaged it so much that your T cells, right, those are your soldier cells, are now down to 200, leaving you susceptible for infection. Now that you are weakened, normal everyday infections can take over. So skin infections, fungal infections. If you have HIV or AIDS, it's going to be a lot harder to fight off. And it's not AIDS that kill you, it's those opportunist infections that do. Additional risk factors. So for HIV, if we know the mother is HIV positive, we do a big cesarean. We move the fetus out and we start the child on viral medication right away. So as long as we know no infant has contracted HIV during the birthing process. Initial infection. So this can stay hidden not for years but decades. 10 years, 20, some people even 30 years after exposure, the virus will then finally express itself. So that's the incubation period. So if you get a needle stick from somebody who's HIV positive or you think you've been exposed, unprotected sex, start retroviral medication right away. It takes about 72 hours for the virus to actually attach to T cells and start infecting you. So if you're exposed, get treated right away. It's a good chance you'll never contract HIV. Full-blown AIDS, of course, only when your T cell count is below 200. Diagnostics. So we have the quick and dirty tests. Right? So that one, get your results in 10-15 minutes. It could be saliva or blood. And then if you get a positive, then we'll confirm it with the more expensive one, the ELISA. So here they're actually sequencing DNA, viral DNA, to see if it's in your body. Western blot test is another one, or you can do the antibody test. So you need two to three to confirm the results. And then if it is positive, we'll start you on your retroviral medication. So HIV is a retrovirus. So what that means is you have healthy human DNA, and what the virus will do is it'll splice in a segment of virus DNA just in the middle of where your normal DNA is. So anytime you copy your DNA, now you're going to copy a little piece of that viral DNA every time T cells replicate. So now you have virus DNA. You don't even need a virus particle, right? All you need is that DNA to be inserted into your healthy DNA. So if you take a look at Magic Johnson's blood, you will not see any virus particles. If he stops taking his medication, this DNA, this viral DNA, will start expressing and he'll start making virus particles. So you don't really get symptoms until you're pretty near full-blown AIDS. So a lot of skin issues, you'll get different cancers, skin cancers that develop now because your body's repair and healing mechanism is disrupted. That T cell, that's part of your immune system, immune and repair. So prevention is the key. So avoid risky sexual activity, IV drug use. So that's why needles are free. So they don't want to spread diseases. So a lot of places will give out free needles. You give me a bottle of all your used hypodermic needles that you use for heroin or whatnot, no questions asked. I'll collect your sharps. That way no one wants to use these needles that are dirty, and we'll give you new ones to prevent the spread of diseases. The goal. So we want to delay full-blown AIDS. So the earlier you get treated, the better. We'll know your strain of HIV and the best way to suppress it. If your partner's HIV positive as well, you'll still need to use safe sex practices because they could be different strains of HIV. All right, hepatitis. So hepatitis A, San Diego's kind of notorious for hepatitis A. So initially for Chipotle, if you recall Chipotle's food scare, they weren't washing their vegetables correctly. So there was a lot of hepatitis A. So that's foodborne, waterborne, and if your vegetables aren't cleaned, then you might catch hepatitis A. Currently, the homeless issue and the spread of hepatitis in that community. There's no place to use the restroom and wash your hands. Hepatitis B and C, so those are gonna be bloodborne. And for D, you cannot have D unless you have B. So if you never gotten B, you'll never get D. So wash your hands, don't eat any poop, and you should be fine. So no blood, no poop, wash your hands, you should be okay. Oh, there's hepatitis B. It was hiding. So for babies born to mothers that are hepatitis B positive, we're going to give them the antibodies. The babies, the antibodies for hepatitis B. If we give them the vaccine, it's not going to work in time. It takes about 2 weeks for the vaccine to work. So for those babies exposed to hepatitis B, we'll give them the IVIG, immunoglobulins. Similar to HIV, if you have blood and bodily fluids being exchanged, you have a high chance of spreading HIV. Certain areas of the world are very close quarters, so a lot of hepatitis. And then if you're hemophilic or get blood transfusion, they used to not screen for it. So in the before the 90s, I don't think they screened for it. So since hepatitis attacks the liver, so it'll damage the liver, might lead to cirrhosis, which is hardening. And then since it is a virus attacking your DNA, it can then lead to cancer. So jaundice or yellowing of the body, that's due to bilirubin. It's a byproduct that your liver is no longer eliminating, getting rid of. So it builds up in your body. So your eyes will look yellow. The skin will also have a yellow appearance. Yeah, so general tiredness. Because remember, your liver is your metabolic center. Now you can't carry out chemical processes. Diagnosis. So we can do a quick test. Just checking for antibodies in your blood. Preventative measures, of course, as a healthcare worker, just use your standard precautions. You should be fine. Get your vaccines, and then here I mentioned the HBIG. It's not the vaccine, it is the passive immunity for it, just in case you're exposed. That way this will fight it off right away. So these are antibodies. Immunoglobulins are the antibody protein. All right, looks like they helped us out a little bit, true or false. So the lines, I guess, are already false. So risk factors are the same for hepatitis B as in C. So are they bloodborne? Yes, they are. Hepatitis A is droplet. So that's water, foodborne. Helper T cells are not red blood cells, they are white blood cells. Precutaneous exposure, so this is skin through puncture wound or needle sticks. Is that? Yeah, through the skin. Yep. Well, viruses and pyrons. We just didn't mention pyron officially in the lecture. Viruses are complex? No, very simple. Remember, all they need is a protein coat and some genetic material, and bam, you have a virus. All right, that brings us to the end of part 1 of microbiology and disease. So we'll get into the bulk of the microbes tomorrow. We'll talk about bacteria, different types of bacteria, fungus, protozoan, and we'll touch on multicellular organisms, the parasites.